Our next topic in 21.8 is denaturation of a protein. Protein denaturation, also something that happens inside our body. But this is typically not a metabolic process. Like when we're doing hydrolysis, it's good. We're taking the protein and we're breaking it down into amino acids that we're going to do something else with. We're going to make some new protein with them. In denaturation, this is more of a destructive process. So this is where we are going to break, our body is going to break some or maybe all of the secondary or tertiary or quaternary interactions within a protein. So the hydrolysis of a protein, this breaks or acts on the primary structure in a disassembly sort of a process. This is something that happens only to the primary structure of a protein. Denaturation is acting on the secondary, tertiary, and or quaternary structure of a protein, disrupting the interactions that give us the secondary, tertiary, or quaternary structures. When we do denaturation of a protein, it changes its biological function because it changes its shape. So this changes the shape and also results in a change to the biological function of the protein. And sometimes this is something that our body does in the immune response. Sometimes we destroy the biological function of something that we don't want in our body so that it can't affect us anymore. So this is affecting the secondary, tertiary, or quaternary interactions. So this is going to be things like our hydrogen bonding. Let's see, we had hydrophobic. Hydrogen bonding was part of the secondary structure, hydrophobic, hydrophilic, and sulfide bridges, which was part of the tertiary and quaternary structures. So this acts on those types of interactions. There are a lot of different causes, a lot of different things that cause denaturation. A very common one inside of our body is heat, like running a fever. Heat tends to break hydrogen bonds, which is going to act on the secondary structure, the alpha helix and the beta pleated sheet. And it also breaks hydrophobic interactions. So that would be part of the tertiary and quaternary structures. Another thing that denatures proteins are acids and bases. This also breaks hydrogen bonds. Chemicals. Specifically organic chemicals like volatiles, very stinky organic chemicals. This breaks hydrogen bonds. Heavy metals. Some of the metals that are heavy in terms of having a high atomic mass, like lead, poisonous, toxic, they break disulfide bridges. And then last but not least, and this isn't a totally comprehensive list, agitation, which is just like stirring it, shaking it, something along those lines. This breaks hydrogen bonds and also hydrophobic. So there's a lot of different ways to denature proteins.